So I'm from Autonomic Neuroscience Department and Geriatric Gerontology Institute. So in the elderly, late elderly, the many problems increase, such as weakness of the bone and muscle, or frequent urination, or like that. And autonomic nervous system innovate variety of tissues and organs. And when somatic tissue like skin or muscles are stimulated, the information goes to the central nervous system and reflexively affect the autonomic nerve activity. This is called somato-autonomic reflex. And as Dr. Ku mentioned, my mentor, Dr. Akio Sato, published when he retired such a review book, and he suggested this somato-autonomic reflex is the basis of acupuncture's effect on the visceral functions. So today I will talk about our recent studies related to somato-autonomic reflex and skeletal muscle functions. My talk will be three-part. First, somatosympathetic reflex affecting the muscle force. And second, changes in the sympathetic muscle force regulation with age. And these are a basic study on animals. And third, effect of acupuncture-like stimulation on chronic neck and shoulder pain. This is clinical study. So, first I'll talk on first part. This is schema about the nerve innervation of the hind limb muscles. There is, of course, motor nerve innervating and produce muscle contraction, and afferent nerve convey information, sensory information to the central nervous system. And additionally, there is sympathetic nerve. In the case of hind limb, only sympathetic, not parasympathetic. Sympathetic nerve. So many sympathetic nerve are included in the hind limb muscle nerve. This is showed by Professor Schmidt. In the case of lateral gastrocnemius muscle nerve, more than 40% of number of 40% is sympathetic postganglionic fibers, efferent fibers. So many sympathetic exist. And it is well known sympathetic nerve innervate arteries of various tissue, but in skeletal muscles, it is historically shown sympathetic nerve not only innervate artery, but also skeletal muscle fibers. And recently, this is a recent study, histological laser microscope study. This green is adrenergic sympathetic fiber, and red is neuromuscular junction, cholinergic receptors. So most of neuromuscular junctions are innervated by sympathetic nerve. And in vitro neuromuscular nerve muscular preparation, when sympathetic nerve are denervated, after 7 days, muscle cross-sectional area reduced by 20%, and contractile force, especially those by nerve stimulation, was reduced. However, physiological significance of muscle sympathetic activity on skeletal muscle force is not still clear. So we hypothesized the somato-muscle sympathetic reflex associated with contraction of skeletal muscles help maintain their contractile force. So to clarify this hypothesis, in the first part, we did 5 experiments under anesthesia. We estimated reflex pathway. One is efferent pathway, that is lumbar sympathetic nerve, LST. This is specific pathway going to only sympathetic nerve contained, going to the hind limb muscle, and also effect of cutting efferent and also effect of cutting afferent, those are root, and cutting central nervous system, spinal cord at cervical level, 3 cut experiment, and then we recorded sympathetic nerve activity, whether it reflex activation occurs. And last, we stimulated cut peripheral end of lumbosympathetic trunk if they cause the changes in muscle strength. And this is a classical, simple experiment. Under anesthesia, we isometric force produced by motor nerve stimulation was measured by thread connected to rod cell. The hind limb was fixed, and stimulation applied to intact tibial nerve. The strength is twice the motor threshold. So it means activate thick fiber like motor nerve, but not thin fiber like sympathetic or small thin sensory afferent. And we stimulate burst stimulation, 0.2 millisecond, twice the motor threshold, and burst 100 Hz, 10 impulse. This causes tetanic contraction. The tetanic contraction repeats every 1 second, 10 times, and then avoid fatigue. We have rest, so we repeat this 10 tetanic contraction 3 times. So altogether 30 tetanic contraction as one set. And we measured the force generated before and after LST ipsilateral to the force measuring muscle was examined. Cut. And this is one example. And 30 contraction, maximum contraction decreased after acute LST cut. 20 minutes after it's decreased. And this is a superimposed contraction curve. Shape is same, but peak amplitude reduced slightly. And when summarized in 6 animals, the reduction is average 10% of the initial force. So, cutting the lumbar sympathetic trunk, LST, decreased tetanic force amplitude by 10%. And then, in another experiment, this is same, this is efferent cut. And when spinal cord was cut at the cervical level, similar decrease. And those are root afferent pathway cut, similar 10% decrease. So any estimated reflex pathway caused 10% reduction of the force. And for this experiment, we recorded this gray-ramus postganglionic sympathetic nerve activity going to the hind limb. And in the central nervous system intact condition, there is early excitation and late excitation. And when the cutting the spinal cord, only very early excitation appear. So contraction of hind limb muscles induced somatosympathetic reflex, spinal reflex, and also supraspinal reflex in the lumbar sympathetic activity. And finally, we stimulated cut peripheral end of the LST electrically, and so it increased, then increased force amplitude. And this effect is depending on the stimulus frequency. 1 Hz stimulation had not clear effect, but 5 Hz, 10 Hz, 20 Hz of sympathetic stimulation increased the force amplitude. So, in conclusion of the first part, when motor nerve activate and muscle contract, then afferent nerve, it is known, group 3 and 14, afferent activate and maybe it produce sympathetic reflex, spinal and supraspinal reflex, and increase the sympathetic activity, affect probably neuromuscular junction, and increase the contraction force. So there is a feedback system between muscle contraction and sympathetic nerve activity. So if this feedback mechanism declines, it could cause muscle weakness. So in the next talk, next part, we examine the feedback relationship in aged animals. Second part, changes in sympathetic muscle force regulation with age. I talked about young rat reserve. This is about 100 to 300 days old animal. Then we also studied similar experiment on aged animals, about 1,000 days old. That is survival to only 20%, so very, very aged, maybe 80s in humans. And this right axis shows the relative weight of the triceps muscle, calf muscle. In young rat, that is 0.6% in both male and female. This is the male case, but male and female percentage body weight was same, 0.6%. And aged animal, that percentage decreased about 0.4 or 0.2 variety. And using such animal, at first we did LST cutting sympathetic nerve. Then cutting sympathetic nerve in young animals caused 12% reduction of the force, but in aged animal, the reduction was only 6%, so significantly reduced the contribution of the sympathetic nerve. Sympathetic contribution to tetanic force became smaller with age. Then next, subgroup analysis. This includes both male and female, so it divided. Then the significant reduction was only observed in male, but not female. And male rat, as I showed, relative muscle weight have variety. So we found the contribution of tetanic force of the sympathetic contribution clearly very significant correlation between the muscle atrophy. So less muscle mass, smaller the sympathetic contribution. And this is a stimulation experiment. During stimulation of the LSD, in the case of 10 Hz stimulation, young and aged animal both increase the force. However, if we studied on the frequency dependent in aged animals, 5 Hz had not significant effect, and 10 and 20 Hz had significant effect. So it seems frequency threshold to increase tetanic force became higher in age. And further, interestingly, we found this experiment. Before stimulating the tibial nerve, we stimulated LSD for 1 minute solely. And in the case of young animal, the sympathetic stimulation not clearly affect the muscle tension, muscle tonus, very small, but in the aged animals, most aged animals showed clear increase in muscle tonus only by sympathetic stimulation. So sympathetically generated muscle tonus was augmented in aged rats. This is also very interesting. And I checked the contribution of adrenergic receptors, alpha blocker phentolamine and beta blocker propanol effects, and found both alpha and beta adrenergic receptors are involved in an increase in tetanic force modulation. And in the case of tonus increased by sympathetic stimulation, alpha but not beta adrenergic receptors are involved and increase in muscle tonus. This is only decreased by alpha blocker but not affected by beta blocker. So in the summary of the second part, with aging, the sympathetic contribution to help motor nerve function, to help tetanic force generation decreased. However, sympathetic generation of resting muscle tonus, alpha adrenergic involvement of the tonus increased. So such changes may be related to, this is related to maybe muscle weakness, and tonus increase may be related to muscle stiffness or muscle pain. So last part, I will talk on effect of acupuncture-like stimulation on chronic neck and shoulder pain. As introduction, this is a memorial paper of Dr. Akio Sato showing morphine blocks transmission of the afferent C fibers at the spinal segmental level in anesthetized cat, recording cardiac sympathetic nerve efferent activity and stimulation of forelimb or hind limb electrically cause A and C2 reflex, early A reflex and late C reflex, because somatic nerve contains fast conducting A fiber and slowly conducting C fiber. And C fiber includes some deep uncomfortable pain and related to chronic pain. And morphine injected intrathecally to lumbar area only suppressed C reflex of the induced by hind limb stimulation, not forelimb. This is interesting. Morphines, one of morphine mechanism is afferent C fiber transmission block in the spinal segmental level. And my experiment showed similar results observed by acupuncture-like stimulation. I recorded in rat cardiac sympathetic nerve and stimulate hind limb nerve and cause A and C reflex. Then I applied acupuncture-like stimulation of such many microcones just to stimulate the surface of skin. Then only not A reflex was affected, only C reflex was depressed, so similar to morphine. And this effect is blocked by opioidergic antagonist, naloxone. So recently, we analyzed 2-week intervention with this acupuncture-like stimulation for chronic pain. This called somalezone. Small company made such a special acupuncture. This is 180 microcones on the surface of 7 millimeter, and this can use as self-care. So participants are 12 men and women with chronic more than 3 months continuing chronic neck and shoulder discomfort, age is variety of age 27 to 62. Attaching the microcones by themselves every day, and area they feel stiffness, and up to like this, up to 8 patch that's applied, and 12 hours daily for 2 weeks continuously. Then we measured subjective and objective symptoms. This is the result of subjective symptom. As subjective symptom, visual analog scale, maximum 10 centimeter. Maximum imaginable pain is 10, no pain is 0. So these people have about 7 pain sensation or discomfort average. And that become, after 2 weeks, reduced to 2. And the case of difficulty in moving sensation, that's 6, and reduced to 2. So very clear, significant effect. This is subjective, so also analyze about objective symptoms. As objective symptoms, physical therapists measure joint movement. This is cervical joint movement, flexion. Flexion and lateral flexion and rotation significantly improved. For example, flexion is very clear. It was 40. This line is a reference range of motion by rehabilitation medicine. So it was very low for 40 degree. It became 60, a normal level. And also measure shoulder joint movement, flexion, extension, internal rotation was significantly improved. And shoulder girdle movement, flexion, extension, and these 2 significantly improved. So among 12 movements, 8 movements improved. So we need to analyze mechanism more, but maybe acupuncture-like stimulation relieved chronic muscle pain, possibly by inhibiting the transmission of afferent C fibers, conveying discomfort, and also inhibiting muscle tonus generated by somatosympathetic reflex, as I showed in the first and second part. So this is last slide. Sympathetic nerves innervating hindering muscles are activated in response to muscle contraction, facilitate contractile force. And age-rat feedback became sympathetic nerve and muscle contractility became weak, but sympathetically induced muscle tonus are augmented. The somatosympathetic reflex may be involved as the mechanism to improve symptoms in chronic muscle pain by acupuncture-like stimulation. So thank you for attention. This is my laboratory. Yeah, I thank many people collaborated with this study. Thank you very much.